All right. Thank you. I know you have been looking forward to this video, epic program, Blood Talks, the science of what flows to you. In last episodes, we've been talking about the story of a young man, Stephen, who got married to his heartthrob, Tiwalade, without doing a hemoglobin genotype test. And now came down with a baby with sickle cell disease. Now, do you now blame Tiwalade and Stephen for not checking their genotype? Yes. Should they have checked their genotype previously and found out that they were both AS and AS? Should they have separated? What do you think? Today we will go to that answer to that question. Then you will answer that question at the end of this discourse. Should they be separate? I know a lot of you have found yourself in that similar position where your spouse is AS and you are also AS, and you are also afraid of giving birth to a baby with sickle cell disease. Even in the past, a lot of couples have had to separate because they have discordant genotypes. Should this be the case? Should couple separate because of differences in genotype? Let us answer that question this way. So let's look at Stephen and Tiwalade. The first mistake they made was that they did not even check at all. They should have checked because there are solutions to the problem. Now, what are the options that they have? Now they are married. What option do they have to prevent having another baby with sickle cell disease? Now, or should they stop giving birth to children? Should they go do family planning? No, they don't need to do family planning. I can't tell you categorically that there are now ways that you can prevent yourself from getting sickle cell disease baby when both parties are genotype AS. Now, we will be discussing what is called the prenatal diagnosis. What is prenatal diagnosis? Pre means before, natal means delivery, diagnosis, investigation. So, prenatal diagnosis means investigations that you can do before a child is delivered. Simple. Okay? Now, the prenatal diagnosis cuts across different tests that is being done now to detect the occurrence of sickle cell disease fetus before they are born. And I can tell you that many other things are happening in the science world which you need to know about. Information, they say, is power. Now, conventionally, the prenatal diagnosis that we used to do was a procedure that was done between 10 and 14 weeks of pregnancy. There are basically three main types. One, the first thing is the chorionic villi biopsy. The chorionic villi can be called the young placenta. Now, under specific ultrasound, we can take a sample, a needle sample, a needle put in the abdomen of the woman to pick a sample of the placenta for DNA analysis. That can be done between 10 to 14 weeks, and babies that are going to become sickle cell disease can be identified. Another type of this prenatal diagnosis is called the amniocentesis. What is amniocentesis? Don't mind the big word. It's just a syringe and needle collection of the water that is surrounding the baby. The water surrounding the baby contains certain cells that are peeled off from the body of the baby, from which DNA can be collected and tested for genetic disease. Okay? This is said to be the simplest method of prenatal diagnosis. Now, another method that is available is fetal blood sampling, where also a needle can be passed through the abdomen of the woman, directed to the umbilical cord of the baby, and blood sample from the baby can be drawn for DNA testing for sickle cell. Now, these were the tests that were existing. These have been existing in Nigeria since around 1994. So it's not a new thing. You only need to know where it can be done. But currently, because of the need to stick a needle in and collect samples from the baby, new techniques have evolved. Parental diagnosis, where you can collect blood sample from the mother of a pregnant baby, and part of the DNA of the baby that is circulating in the mother from cells that have crossed from the placenta to the mother's circulation. Those cells, fetal cells, can be harvested and DNA tested. These are new scientific methods. Okay? Now, most beautifully now, we now have what they call the PGD, the prenatal genetic diagnosis. Prenatal genetic diagnosis, which some people also call prenatal genetic testing, is finally in Nigeria. It's in Nigeria at the moment. And what do? If you want to circumvent all the psychological and ethical issues that are surrounding prenatal diagnosis, as in when you have to diagnose a baby for sickle cell in the mother's womb, what do you do with that baby? Do you terminate or not? Those issues have been overtaken by this PGD. Now, in PGD, you just go and do an IVF procedure. Now, in the course of the IVF, when the embryo is being cultured, part of the cell of the embryo will be pretested for the presence of known genetic disorders. Okay? Now, any of the cell that has genetic disorder will not be transplanted. Only eggs that have been tested and are free of genetic disorder. Don't forget that you can be testing for only one genetic disease, or you can test for a variety of genetic disease before the fetus is implanted. And of course, the woman will carry the baby normally and deliver normally after 9 months. Disease-free, sickle cell-free. So, gone are the days when you will say that people who have AS should not marry AS. If the fund is available, there are very sweet options that can be adopted, and women and their husbands will deliver babies that are free of genetic disease. This is Blood Talks, the science of what flows through you. I'm your host, Professor Vincent. I would like you to send in any clarification, any areas you want me to clarify in this discussion. If you need information, please reach out to me. You can like my page and send in your questions. I'll be looking forward to seeing you in my next episode, where we'll be discussing the treatment options for sickle cell disease. Now we'll be looking at details of new treatment techniques that are available, that are even curative for sickle cell disease. Stay tuned. Thank you very much.